In this video, we will explore through the mechanisms of DNA replication in prokaryotes and eukaryotes and how they ensure that this molecule is conserved and passed on in generations to ensure functioning and survival of species of eukaryotes but also prokaryotes. The process of DNA replication entailed certain core processes that are universal between organisms and are essentially the same between prokaryotes and eukaryotes. The differences arise from the quantity of DNA that needs to be replicated, which comes in the seemingly vast difference in the length of DNA between eukaryotes and prokaryotes. The differences arise also between the proteins and enzymes involved in performing DNA replication, which in eukaryotes are clearly distinct in sequence and evolutionarily unrelated to their prokaryotic counterparts. This indicates that DNA replication has evolved separately, but its conservation is essential and the most effective way to proliferate and maintain life. Looking at the initiation of DNA replication. First, we have the origin of replication. DNA replication is initiated at a specific site, which is the origin of replication. In prokaryotes, this sequence is ORC, to which the initiator protein DNA-A binds to specific tandem repeats called DNA-A boxes. Binding of DNA-A to the origin spreads beyond the DNA-A boxes to an adjacent AT-rich region, the DNA unwinding element, or DUE. Due to the size of eukaryotic genomes, while prokaryotes have one origin of replication, eukaryotic DNA replication is initiated in more than 10,000 sites. This adds a level of complexity to eukaryotic DNA replication, involving the origin of firing. To ensure accurate genome replication and prevent replication mismatches, coordination of signals is paramount. In prokaryotes, replication begins at a single origin point, proceeding bidirectionally. In contrast, eukaryotes employ a more intricate process, separating origin licensing in G1 from origin firing at the start of S phase. Origin licensing involves loading the MCM2 to 7 helicase onto DNA by the origin recognition complex, or ORC, while origin firing requires the assembly of the replisome on the MCM2 to 7 helicase. This sophisticated eukaryotic approach, coupled with the recruitment of coactivator proteins CDC45 and GINs and CDK phosphorylation activity to coordinate DNA replication with the cell cycle and ensure the genomic content can be maintained between cell generations, avoiding genome instability from aneuploidy, which is an unequal number of chromosomes. Next, we have helicase recruitment and DNA melting. In prokaryotic DNA, DNA-A binding causes local unwinding and separation of the strands, which creates a loading site for DNA-B. DNA-B is an ATP-dependent DNA helicase that unwinds double-stranded DNA. It is a hexameric ring and causes DNA unwinding by steric exclusion, which is the breaking of hydrogen bonds between base pairs to translocate one strand from the other. To load DNA-B onto the DNA requires a helicase loading protein, DNA-C, to break and reform the hexameric ring around the exposed DNA strand. As the DNA helicase unwinds the DNA strands, they have a tendency to reanneal, which is counteracted by single-stranded DNA binding proteins, or SSBs, that coat the DNA strands to prevent reannealing and to protect it from possible nuclease degradation. The DNA helicases are involved in creating the phenomenon of replication forks as they go in a bidirectional manner. The key difference is in the replicative machinery utilized, which arises from DNA-A in bacteria being the only origin binding protein, while in eukaryotes there are 6 oct proteins for that function. The variation is also present in the DNA helicase used, where in eukaryotes there are several different types, with the most prominent being the MCM complex. The next stage of DNA replication involves polymerizing new polypeptide strands, which is carried out by a group of enzymes called DNA polymerases. E. coli has 5 DNA polymerases, while at least 15 DNA polymerases have been found in humans. For DNA polymerases to start polymerizing a new DNA strand, They require a short nucleotide sequence to bind onto DNA, which is known as the primer. This requires a specialized polymerase, known as primase, to get them started with a primer. Primase synthesizes a short RNA primer on the template DNA that is then extended by a DNA polymerase. The differences arising in the priming of DNA between the primase used by the cell, which is DNA G in prokaryotes, and Pol A or primase in eukaryotes. Nucleotide polymerization by DNA polymerase in prokaryotes begins when DNA polymerase III, which is binding to a primer, adds nucleotides in sequence to it along the replication forks in a 5' to 3' direction. This means that nucleotides are added to the 3' end of the primer. Due to the DNA strands being antiparallel and DNA polymerases only be polymerizing in a 5' to 3' direction, this results in one strand formed from continuous polymerization of the leading strand, while another discontinuously on the lagging strand, which consists of Okazaki fragments. Okazaki fragments are the short sequences of synthesized DNA formed discontinuously on the lagging strand by DNA polymerase, binding to the primer. The main topological difference between prokaryotes and eukaryotes here is the length of these Okazaki fragments, which arises from the level of priming. In eukaryotes, each Okazaki fragment is around 1,500 base pairs, 150 base pairs, while in prokaryotes it's roughly 1,000 base pairs. Also, the larger number of eukaryotic polymerases makes the cell better adapted for the complexity of the larger eukaryotic genome, with more specialized function, such as performing nucleotide polymerization in the presence of damage or gaps in the new DNA. The large number of repair polymerases within eukaryotes highlights the efforts by our cells to protect and maintain the genetic information encoded in our DNA, which is not present in prokaryotes to the same extent, if at all, as genetic instability allows for variation and the evolution of bacteria. The removal of RNA primers and ligation of Okazaki fragments. In prokaryotic DNA replication, the removal of RNA primers and ligation of Okazaki fragments rely on DNA polymerase I. This enzyme, equipped with 5' to 3' exonuclease activity, hydrolyzes the RNA primer while synthesizing DNA, facilitating the replacement of RNA with DNA. The process concludes with DNA ligase sealing the DNA fragments. In contrast, eukaryotes utilize a distinctive mechanism. RNase H, a specialized ribonuclease, eliminates the RNA primer paired with DNA. Additionally, the replicative DNA polymerase displaces the residual primer, forming a flap structure. Recognition and end excision of this structure involves flap endonuclease FEN1, and the resulting nick is sealed then by DNA ligase. The coordination of this process is guided by the eukaryotic sliding clamp pCNA. Processivity is the uninterrupted polymerization of nucleotides, which is a crucial aspect of DNA replication. In prokaryotes, DNA2 achieves processive synthesis using a sliding clamp protein, the beta clamp. This clamp subunit forms a topological link to the DNA template, ensuring the polymerase remains connected during leading strand synthesis. However, multiple rounds of clamp binding and release are necessary during the lagging strand synthesis for Okazaki fragment synthesis. Eukaryotic replication mirrors this, with a sliding clamp pCNA orchestrating these processes. The sliding clamp allows for processivity in DNA replication, referring to the ability of DNA polymerase to continuously synthesize nucleotides without interruption. This interruption activity is essential for efficient and accurate replication in accordance with the cell cycle and cell division. Prokaryotic cellular replication undertakes the same processes as eukaryotes, but in a simpler format. Strand replication is initiated at a single site and is propagated by DNA polymerase III primarily, closely associated with the beta clamp that is orchestrating the synthesis. Eukaryotes introduce complexity with multiple origins necessitated by larger genomes, involving more functional units, for example polymerase activity, initiator proteins, helicases, and pCNA to form the replisome and coordinate the rate and efficacy of DNA replication. This added complexity in eukaryotes becomes crucial for accurate DNA replication, a fundamental process vital for cellular function and integrity, intertwined with the cell cycle. In a clinical context, these intricacies significantly impact cancer diagnostics and therapeutics. Chemotherapeutic strategies targeting the replication machinery aim to halt abnormal cell growth, underscoring the clinical relevance of understanding these differences. Biomarkers like MCMs play a pivotal role in identifying and assessing abnormal cell proliferation, offering valuable insights for disease diagnoses and informed treatment decisions. Thus, comprehending the nuances of DNA replication holds substantial clinical importance, shaping strategies for disease management. Thank you for tuning in on our video, which really hoped to contrast the differences in DNA replication in prokaryotes and eukaryotes. Stay tuned and please subscribe, like the video, and leave any comments on anything we can do better or any new topics that you would like to hear about. We will shortly be releasing videos on DNA repair strategies and also the cell cycle to um, to keep it within the same topic and give you more insight into this field. I hope now you understand the process of DNA replication in prokaryotes and eukaryotes, and we'll catch you in the next one.